Hi, my name is Michael Spedding, President of Spedding Research Solutions. I'm celebrating 52 years of experimentation and 10 years of Spedding Research Solutions, where we've got a drug in clinical trials for motor neuron disease, ALS. And I'd like to show you how our society works, how our company works, and the possibilities for interactions about multiple areas and our points of interest. And I'll just share the screen. So, Spedding Research Solutions does research into impossible diseases based on my experience in 3 pharmaceutical companies in the research committees for 35 years, seeing every way drugs can pass or fail, but also with some good new ideas in motor neuron disease, long COVID, aging, neuroinflammation, particularly based about metabolomics. So I actually retired from Servier and left Servier with a big research grant. So we set up Spedding Research Solutions, which can then interact with pharma, directly with universities, biotechs, and CROs. We do collaboration and fee for service, and also directly consultancy, looking at new research areas, and we've been pretty effective in getting grants, an enormous number of publications, which we co-authored, and I'm very proud to be associated. In fact, I'm in the 3,000 most cited scientists. There's only 5 companies listed in pharmacology: GSK, Pfizer, AZ, Johnson & Johnson, and SRS with 2 employees. So we look at, we develop patents, get compounds, but we're very effective in raising what money we can raise is spent very judiciously, and we can get back money on credit ample research. And the strategy is no debt in the core company. That means that our deadlines are our own deadlines. And so this 10 years of activity has given us quite a lot of progress, particularly in ALS. And the way that we set up the ALS program was by collaborations. Initially with Servier, then, but also particularly with Jean-Philippe Loeffler's group in Strasbourg, where I had a fantastic collaboration. And Alex Henriquez at Neurosys, who's now at Eurosys, was also key in that, was our project leader also, who was employed for a time by me, but working in Jean-Philippe Loeffler's lab, and did some fabulous work, as you'll see in the presentations. Gail Westland's fantastic for the scientific organization. We've got academic collaborations around the world. We link with SAT Connectus, which we share, which is the bridge between universities and INSAM, which we contract with and share IP with. And we've raised grants, and we've now, with Brad Turner's group at the Florey, doing investigator-driven trials for ALS with Ambroxol. This is the way we're set up, with collaborations for a whole variety of different areas. And so we've got a particular rationale for using evolution, which you see the film about. Human evolution means people are quite different from animals, and with a different metabolic evolution, which gives us a risk for certain diseases. And Kaitovich has shown we evolved by making mitochondria more efficient and by using lipid metabolism. And my friend Jean-Philippe Loeffler in Strasbourg had showed that energy metabolism was a key issue in amyotrophic lateral sclerosis, and lipids were key. And so we set up a collaboration with Servier, 3,000 lipids, and in these experimental animals, and found superoxide in the superoxide dismutase model. Metabolic and transcriptomic analysis showed that ceramide and glucosylceramide was a critical role. And we found from that glucosylceramidase being critical, that Ambroxol could interact with it in a very specific way. And we got patents, often do drug designation, and the phase 2 in trial. And it's the only drug to have been effective in 3 major models, critically affecting neuromuscular junctions. This is the group, tremendous. So we're really probably one of the most effective groups in the world about interacting worldwide. And we've raised $2.5 million in grants over 6 years, but that's all also by all of the partners. about 500,000 came through SRS. And thank all these partners very much. One particular issue is that why we're looking at the next 50 years of research, which is the complexity of glycosphingolipids, because 5 sugars can give you 2 billion structures linked to also the 400,000 structures of lipids such as ceramide. So GM1 is actually a critical factor. It's 17% of the brain glycosphingolipids. It's neurotrophic, it binds to track B, etc. And so it's a key factor, which was also key in evolution because it's also a target for viruses, and it maintains neuromuscular junctions. And therefore, we've at this Congress, we can see Elena Chiricozzi is presenting specifically on it. And it'll be in our presentations on the Wednesday afternoon on the ALS program. And the sialic acid in GM1 was specifically mutated 2.5 million years ago, and it's probably critical for human evolution. It's a target for viruses, and therefore, by having this mutation, probably early hominins escaped a viral infection, which possibly wiped out the rest of the hominids. And it's actually critical for neuromuscular junctions. So we did a complete metabolomics. So these are the papers that we published, and validated also by Genzyme, and between ceramide and glucosylceramide, a critical node. I'm not going to, it's in our more detailed presentation on ALS, but ambroxol, a very specific effect there, highly reproducible in all studies. And inhibition of synthesis of glucosylceramide exacerbates denervation in ALS, and inhibition is protective in ALS and also in helping denervation recover. And lipids are really markedly changed. So also, we've been able to look at the cholera toxin will bind to GM1. And so we can actually see the neuromuscular junctions with the glycosphingolipids, and cholera toxin lost the very first stage of ALS. And this is probably because the glycocerebrosidase GBA2, which is the non-lysosomal one, is specifically increased in the spinal cord right at the start of the disease. And ambroxol inhibits GBA2 as a chaperone for GBA1, which means it can probably work both in Parkinson's, whereby you need an increased GBA1 activity in lysosomes, and in ALS, where you need to inhibit this GBA2, breaking down GM1 and destroying neuromuscular junctions. That's our theory. So, and also, so very quickly, once it was passed with SARS came along, we were able within just months to show that ambroxol would inhibit the viral load of SARS-CoV-2 replication at the same concentrations. It would increase neuromuscular junction formation in vitro. Right, so we're very pleased about that, and it means that the two effects are probably work at similar concentrations by GBA2, and we've been able to show with Rushka Pereira that it's a critical node also for viral release. So ceramide glucosylceramide link is critical for viral infections and also for neuromuscular junctions, and we hope for ALS. So this means that there's a big, shows how reactive we can be, therefore the mechanisms for COVID, long COVID and neurodegenerations may be linked. This is a big issue for the European Union, and so we got actually a grant from meetings to just discuss how crucial this was, and it's a major issue which we've been working with, and we're now linking with Recover, which is the $2 billion US long COVID initiative from NIH. And the title of the grant was Sphingolipids as Crucial Links Between COVID-19 and Neurodegeneration, with worldwide experts. At present, we think that the incidence of ALS Parkinson's is not increased in a major way by long COVID, but the enzymes involved are very similar, and that the metabolomic effects of long COVID may actually have deleterious effects in old people for Alzheimer's just by reducing their energy capacity in VO2 max. So this is a way forward, which I think is being very quickly worked out, shows how reactive our company can be, looking for major issues for healthcare, And that's because of all the wonderful interactions that are having all around the world, and gives us a possibility also for you to be able to join in some of those. So effectively, Ambroxol, we've patented for neuromuscular disorders, viral infection, dengue. It's got orphan drug designation for ALS, and we're in interactions with the Tony Shapira's group, who are developing it for Parkinson's disease. So it's already actually started clinical trials with us, and Turner particularly, I must emphasize how important our Australian interactions have been for ALS. And then we keep a tight link with the interactions with Parkinson's disease, where the phase 3's just started. And we've got a clinical plan for Australia, USA, UK, where the drug isn't registered. So we may be able to make a business case for that, and also Europe. And then, so those are the ALS issues as an example of what Spelling Research Solutions can do. We've got multiple other approaches, which you can see in the films. You just flash the QR code on your phone and you can follow us in a quiet moment in the meeting or at home, or just go into the Spelling YouTube channel and pick them up. So circuit-based approaches for drug discovery and mental health, where with Servier, we were able to do some groundbreaking work, and now Macron has given 80 million for a new research centres in France based on mental health and the circuit-based approaches which we've pioneered. Environmental pharmacology, prevent us poisoning the earth with chemicals which haven't been analysed pharmacologically, even if they have been toxicologically. Aging research in sports science, look at the film, it's quite amusing. Human evolution, as you've seen, is important. I also have an issue, after all these years of research, about reproducibility, how to control black boxes, and also to look for drugs which are completely different using the black swan approach. And also, because I've been on research committees sent to research centres for giving creativity presentations to young people, how to manage it and how to manage their lives. So I'm at this meeting. I'm very busy as Secretary General, so I won't be at the stands. This is why this film's on. And so I suggest you chase me. I'll try and get here for coffee and discuss and argue. So that's what it looked like. And I'm looking forward to interactions and having potentially further ways forward for our research. So thank you very much.